some of these clones are too, but not these white ones, which tells me that the bacteria in this colony, in this dot, have taken in the gene that I want them to take. And so in molecular cloning, we can stick the genetic instructions for making a protein. So we can stick like the gene for this protein into a circular piece of DNA called a plasmid vector, and then we stick that into bacteria, and the bacteria will make lots and lots of copies of it, and then we can stick it in other bacteria or in other cells and get them to make the protein. But in order to do all that, we need to make sure that our cloning actually worked. So we need to make sure that the gene that we put in actually gets into the plasmid, and then that plasmid gets into cells. And so we can use like antibiotic selection to test if the plasmid got in there, but so if the like vector that it was in got into the cells, then it has the antibiotic resistance gene, and if we have the grow it into antibiotic that it's the complementary antibiotic, then it will survive. But if we don't know if the gene that we put into that plasmid actually got in there. And so there are techniques that we can use that are kind of time consuming, like colony PCR or analytical restriction digest to get an idea if it's in there, and then sequencing to really check. But we have to do some work for all of those things. And well, scientists, we do enough work. So let's make things easier and do screening in addition to selection. So with selection, you're actually like saying, okay, only the things that have some certain feature can grow. So only the things that have the plasmid, so that have the antibiotic resistance gene, can grow. But with selection, what you're doing is you're saying, okay, just show me the ones that have it and the ones that don't. And that way I can kind of like prioritize which ones probably have it inside. And so when we use a combination of these special plates, these special bacteria, and these special plasmids, we can kind of take advantage of this bacterial beta-galactosidase system in order to disrupt a gene that the bacteria need in order to make a blue product. And then if we get our plasmid, if we disrupt that gene, then they can't make the blue thing, and so that tells us that the plasmid is inside because it grew because of the antibiotic, and the fact that it's not blue tells us that our gene got inserted into the region. And so now let's look in more depth into how this works in theory and practice, and the really cool biochemistry going on. So quick terminology note. When I say like we're putting a gene in, more specifically, we're putting in like a cDNA. So basically, more on this in other posts, but a cDNA or complementary DNA is the DNA version of a mature messenger RNA. And so a gene, the DNA version of the protein instructions is a gene. This gets transcribed into messenger RNA and which gets translated into protein. But before this messenger RNA gets translated, it goes in a little editing, and so it has like these regulatory intron regions removed. And so then we want to put a DNA version of this mature version. So this already remove the introns and everything, so we don't have to worry about those introns because the bacteria aren't going to know how to splice them out and stuff. And so we're just going to put in the cDNA, which is the complementary version of the messenger RNA. But we typically still just say like we put the gene in or whatever, but technically it's the cDNA, and we can also stick other things in. So we can just refer to it as an insert that we're putting in. So I'll probably use the terms all like kind of interchangeably. But the basic overview of this technique is that we're going to put that insert, no matter what we're putting in, we're going to put it in a gene in our plasmid. So in addition to having that antibiotic resistance gene for antibiotic selection, this plasmid is also going to have a gene for part of the beta-galactosidase enzyme that the bacteria is going to need to make a blue product in a way that we'll talk about. And when we put the insert we insert it inside. So we make this insertion sequence so that it's inside of that gene for the beta-gal parts, this like alpha peptide. And then, so this is going to disrupt it so that they can't make this blue product. And so how all this works is it's taking advantage of the bacterial, the galactosidase and lac operon system. So basically, beta-galactosidase is this enzyme, so this reaction speeder upper that bacteria can use to break down the sugar lactose. And so they can use beta-galactosidase to hydrolyze or use water to split
lactose, so this lactose into its component monomers, galactose and glucose. But they're only going to want to make beta-galactosidase if they have a lot of lactose and not very much glucose, because they prefer glucose. So if they have plenty of glucose, they don't want to have to, like, they can get the glucose by breaking up lactose, but they have to like go through work to break it down. And so they'd rather just use glucose and not have to worry about making this beta-galactosidase if they don't need to break down that lactose, which either because they have plenty of glucose or because there's no lactose around. So they don't want to waste the energy and stuff to make this enzyme that they don't need. But they want to be able to make it when they do need it. And so they have this cool system called the lac operon. And basically they have the gene for making beta-galactosidase, beta-gal, on this LAC-Z gene. Ahead of it in the bacterial DNA is this promoter, which is going to tell the RNA polymerase to hop on and start making messenger RNA that's going to get made into protein. And so this is going to get made into this beta-galactosidase. But this promoter is going to be hidden normally by this protein called the lac repressor. So this lac repressor is going to hide the promoter so that the beta-galactosidase doesn't get made. But then when you have a lot of lactose around, then this lac repressor is going to fall off. And why is it going to do this? Well, it turns out that this beta-galactosidase can also kind of moonlight, and what it can do, in addition to breaking down lactose, what it can do is if there's a lot of lactose around, some of it can get rearranged by like transglycosylation to form this allolactose. So basically it just changed the linkage a little. And then this allolactose can bind to the lac repressor. When it binds to the lac repressor, then this is going to cause that repressor to kind of like shape change a little, so conformational change, and then fall off. And once it falls off, now there's the RNA polymerase combined. This LAC-Z can get expressed, so the beta-gal gets made. It actually functions as a homotetramer, so 4 identical chains. So this gene has the instruction for making 1 chain, and so that'd be a monomer. But then it can pair up to form a dimer. And then that dimer is going to form a tetramer, and it's actually this tetramer that is the active version. And so why this part matters is because in order to form this active version, this tetramer, it needs this part, the alpha peptide, which is actually what we're providing in our plasmid. So it's going to function as the tetramer. And so we can take advantage of the fact that it needs to do this tetramerization in order to make functional beta-galactosidase. And what we can do is we can actually make it so that only when the plasmid is in there is it able to make this blue product. So in this Bluewhite screening, so basically what happens is you have in the bacterial DNA, you basically have a mutated version of the beta-galactosidase gene, that LAC-Z gene. It's going to be delta M15. And so this delta means it's like missing things. You've removed amino acids. And so that you remove like the very N terminus part of it, pretty much. This part called like the alpha peptide gene. And so this alpha peptide is required for the tetramerization. And it turns out that you can provide this peptide in trans, so like from a different place. So in this case, from the plasmid, and it's able to complement, so we call this alpha complementation. It's able to complement the not having it here. So it can kind of bind on like it was part of it already. And so this part is needed for the tetramerization, not for the dimerization, but the tetramerization is the part that's functional. And so it needs to have this alpha peptide gene made to get this alpha peptide to get this complementation. So now we have a way that we can get bacteria to make beta-galactosidase. And so in the lab, instead of using allolactose, we use like IPVG, which is going to mimic allolactose, and basically it'll bind and make the repressor change shape and fall off, just like you would if you had lactose, or which you can convert into allolactose. But that lactose can also get broken down, and the allolactose can get broken down and stuff. And so it's you have to keep adding it. But the IPVG, we don't have to worry about that. And so it's like a more stable thing that we can put in and get induction of the expression. And so if this sounds familiar to you, you might be thinking of when we do like inducible expression. This is the same sort of thing. And so we can stick our gene, we can stick a lac repressor, use a lac repressor to just like stick that promoter in front of other things. And so we can stick it in front of a gene for making T7 polymerase, and then put a T7 polymerase in front of our gene on our plasmid, and then induce the expression. And so more on that in my post on bacterial growth expression, but it's the same sort of system, except here we're going to be using it in a different way. So this is all cool and all for the bacteria, and it's really useful that they can make this. But these products aren't very useful for us as a marker. So these products don't have what we call conjugation. So they're not going to absorb light. And we're not going to have something that we can see. But they do have lots of OHs, so they're going to be polar, and water is going to like to hang out with them, and they're going to be soluble. And so we want to make it into something that is going to be colored and insoluble, so it'll like precipitate out. And so how we're going to do this is that we are going to, we can actually change the molecule. So it likes to break down lactose, but it mostly cares about this galactose part. So if we can put something else instead of this glucose, we can put something that's going to allow us to get a colored product. And so we can make these variations in order to get a colored non-soluble product that's going to give us a signal of the bacteria making or not making the product. And so if you're curious about why the whole conjugation thing matters, basically light is going to be made up of little pockets of energy, called photons. Atoms are joined up by sharing pairs of electrons. These electrons, much more on this in other posts, but basically these electrons can get excited, and they can get excited to like a higher energy level if they like catch a photon of a wavelength that they really like. And it turns out that so they can only like catch in wavelengths, they can only get excited by light that perfectly matches or almost perfectly matches the energy levels between like different electron states. And the key thing is that different molecules will have different like frequencies of light that they can absorb. And when you have this thing called conjugation, so when you have these molecules, normally they share like pairs of electrons, but when you have like a conjugated system, such as like an aromatic ring, what happens is these electrons are going to be like sharing in kind of like this communal electron sharing thing. And this is going to decrease the amount of energy that they need to get excited. And because the color of light that you see is going to be opposite of the frequency of the light that's absorbed, then you are going to see a colored product. And so the classic one for this is XGAL. And so basically it gets hydrolyzed into galactose and this other thing. And then this other thing can actually like dimerize and get oxidized to form this blue product, which has this extensive conjugation. So when you see a lot of like alternating double and single bonds, that's kind of indicative that there's likely conjugation going on. So this like resonance stabilization or electron delocalization that's going to allow it to absorb visible light. And it's going to absorb low energy, low frequency, long wavelength light. So what you see, so because it's stealing that part of the rainbow, you're going to see the high energy, high frequency, short wavelength blue light. And so this is why this product is blue. But it turns out this like original, like this XGAL isn't like the only product, and it's not even like the best. So it can be kind of hard to tell how blue things are. So in our lab, we actually use this Bluewhite, which has slightly different structure, it's same idea, but it's going to give you a darker blue product. There are other versions that'll give you like a magenta or a salmon color, various different colors that you can get, all based on the same idea of sticking something onto the galactose that is going to then pair up once it's freed and form a conjugated product that's going to absorb light, and that doesn't have all those OHs that the sugars had. So it's going to be insoluble and precipitates out. And so you're going to get this colored precipitate inside the colony that's going to really let you see it. And so this makes it a pigment as opposed to a dye, although technically we often use the terms interchangeably. Technically a dye is something that's like soluble and a pigment is something that is like insoluble. So this would be a pigment product that's made. Sometimes you'll see blue-white screening actually used in reverse, where like blue is okay, because it's telling you that you've added in this alpha peptide gene as sort of a marker that way, without having like an insert in it, like if you had your gene somewhere else in this plasmid. So there are different variations of blue-white screening, but this is the type that we use, at least in my lab. The white colonies are going to have your insert and the blue ones don't. And so you're going to want to move forward with the white colonies and leave alone the blue colonies. And also remember, it's critical to remember that you have, basically it tells you that there is an insert there, but not necessarily that everything is spelled okay. And so you would, if you want to be really sure, you're going to want to do like send your clone for sequencing. But at least now you have a better idea that there's probably a plasmid or probably an insert in there. And so hope that helps you weed out so you can reduce the amount of work that you have to do in order to get to the really fun stuff playing around with the protein products. So in our lab, we're typically using this as part of our like insect purification, insect cell expression things. So the basic idea with like the baculoviral expression vector system, or BevZ, is you have basically, you do the beginning part, you have to like clone it into bacteria, and then, so this is just like cloning bacteria. We use like DH5 alpha bacteria and get these products, and then we actually like sequence these. So we're not doing the screening at this point. We do it in the later step. So at this step, we're actually going and we're actually doing like sequencing and actually checking that the sequence is actually okay, which is why we're less stringent later when we're just saying, okay, it's white, it's probably okay. Although we typically use, like we do try a few because sometimes there could be problems. But then what we do is we use this to make this backmid. So this was just happening in like a plasmid, so just for bacteria, but we make this backmid, which can basically grow in bacteria or in insect cells. And so it can be used, we can get it to make the insect cells make a baculovirus, so a virus that infects insect cells and gets them to make our protein. But the key thing is that we use this blue-white screening in this step. And so we have this acceptor backmid. So basically our gene, because we do the subcloning in this other, this normal plasmid. Then we want to stick it into this baculoviral DNA to make this backmid. And so when we do this, what we do is we actually, it uses this like, it uses this recombination to get this into this. But basically the recombination sites are basically inside of the LAC-Z gene, and so we put our gene in there, and this is going to then disrupt the LAC-Z so that it can't make the product, and so this is how we do our screening. So this only works with certain bacterial hosts and plasmids. So these are some of the hosts and some of the plasmids, and you can just like Google it and it'll tell you if it's yes or no. But that you also have to make sure that you put the right stuff in the plates. So it's not enough to have the ability to make the product if you don't put in everything that you need. And so I found this in my notes from a few years ago when I forgot to add the blue gal to the plates. And then I made it fresh a few days later. And so, but it's hard to know if in the beginning, there's also remember you need the IPVG and stuff. But sometimes it's like, because it actually takes a while, that's another thing. It takes a couple days for the blue color to start to show up. And so you might initially think, oh yay, they're all white. But then the blue color starts to show up because they need to be able to make all that stuff, like all the beta-galactosidase and stuff in order to convert the product. So if they're all white though, that can be indicative that you forgot to add something to your plates as well. So be aware of that. But yeah, so this is an example. We have these white colonies and these blue colonies, and we go forward with the white colonies. With another thing about these plates is because the dye is going to be or pigment is going to be light sensitive, you're going to want to we wrap the plates in foil, and so to protect them from the light. Also, the blue gao, it's not water soluble, so we typically dissolve it in DMSO right before we use it, and protect it from light beforehand. So those are just some technical notes, and hope that was helpful, and happy cloning. And here's that basic overview, but remember to make sure that you know what type of system that you are using.